Ladies and gentlemen, welcome to the third day of our conference, the first International Synthetic Biology and Biosecurity Conference in Africa, organized right here in Kampala, Uganda, the Pearl of Africa. For the next session on renewable energy, I'm going to be the chair. My name is Ian Peter Busurwa. My training is in biotechnology, but my interests are in policy and communications. So I'd like to take this opportunity to introduce renewable energy and thereafter introduce the very first presenter. So renewable energy is energy that fuels human activities and that is collected from renewable resources, which are naturally replenished on a human time scale. Examples of renewable energy are sunlight, rain, waves, tides, wind, and geothermal heat. All these are carbon neutral sources. Microbial fermentation is one of the most effective ways to produce biological energy through metabolic pathways. Synthetic biology deals with the systematic design and the formation of metabolic pathways, new biological components such as enzymes, genetic circuits, and cells. Therefore, the objective of this session will be to give an insight into the recent advances and opportunities in synthetic biology will make in the renewable energy resources, especially in Africa. So at this particular point in time, I would like to introduce our first speaker, who will be presenting on, the title is SynBio in Africa, a futurist's perspective. And the presentation will be delivered by Andrew Hessel from the Genome Project, right? Andrew Hessel is the President of Humane Genomics, an early-stage company developing synthetic viruses for animal and human health. He is also the co-founder and chairman of the Center of Excellency for Engineering Biology and the Genome Project, right, the international scientific effort to engineer large genomes, including the human genome. He is a former distinguished research scientist at Autodesk Life Sciences. His goal is to help people better understand and use living systems to meet the needs of humanity. Ladies and gentlemen, join me in welcoming Andrew Hessel. Andrew, the floor is yours.
So much. If you can hear me okay?
Yes, we can hear you.
It is an absolute pleasure for me to be here today and speaking with you. Good morning. I just want to point out it has been 17 years since the very first synthetic biology meeting, which was done in 2004 in Cambridge. I couldn't make it to this meeting because it was full. They simply didn't have any more room by the time I learned about it. But this meeting planted seeds that went globally, and it's part of the reason why we're gathered here today. So I just want to point out, this is, you are making history for Africa now. You are building the future. And I just want to thank the organizers, particularly Dr. Jeffrey Oteem, the sponsors who helped make this happen, the organizing committees that pulled this all together, and of course, each and every one of you for attending this and building the community in Africa. This is really a keystone event. For those of you who don't know me, I sit at the intersection of biology and computing. My first love was computers, but I realized I didn't care about them very much. I needed to move into life science. What my perspective, though, is that a cell phone really isn't that different than a simple cell like a bacterium. We know cell phones. We built every bit of our electronic architectures from scratch. We have the stories, we have the people, we have the specifications, we have the knowledge and experience. But to build the physical layer, the components, which we started to put together into circuits and ultimately integrated circuits in the 1970s and beyond, forming computers which linked together, form networks, and ultimately the cloud or the internet. Biology is very similar. It's built up from proteins, from very simple systems, forming biochemical reactions that put together make metabolism that ultimately makes cells, simple or complex. And cells can be networked together to make tissues, organs, full organisms, and ultimately form part of an ecosystem. But there are giant differences between these systems as well. So I just want to be clear. One works in bits, the other works in atoms. One's based in silicon, one's based on carbon. One works in a wet environment, the other one needs to be dry to operate. And of course, one needs to, with life, we didn't build it. We have to figure out how it was built. We have to reverse engineer it. But probably the biggest difference between a cell phone and a cell is that a cell can reproduce. One becomes two and keeps growing exponentially. If our cell phones start to do this, we have a real problem. So synthetic biology, I've watched it emerge over my career, being this intersection of computation, robotics, and life science. And the first intersections, the first overlaps, gave rise to the field of bioinformatics that I worked in, high throughput and lab instrumentation, and more recently, artificial intelligence and machine learning. And it's really this inner core that has come together to create the field of synthetic biology. And it is remarkable, and it is nothing like what we had in the past. So this is changing very rapidly. At the core, though, life is code. Genomes are essentially organisms once fooded. And most of my life has been involved with this very simple circle, this cycle of taking an organism like you see at the top, it doesn't matter which organism, extracting its genome, running it through various sequencing technology that's available at the time, and ultimately getting a file on a computer that represents a gene or genome. And this has been, this work started over 40 years ago with DNA sequencing technologies, and it's only accelerated as long as I've been in the field. What's changing, though, is now we're starting to go from that file that on a computer, and now we have machines that are essentially synthesizers and printers that are able to now print out proteins, which we've been doing for a long time. More recently, print out metabolic pathways. And since 2002, when we made the first synthetic organism, a virus, we've been making full organisms, both natural and unmodified. And as we go around this cycle, this engine, it gets faster, better, and cheaper with each revolution. But I have to tell you, you guys are not late to the table, because it's only made a handful of revolutions, but it will be so much easier for you to get up to full speed, because the equipment, the tools, the technology, the understanding, the financing has gotten so much better in the last few years. My company is called Humane Genomics. I co-founded it in 2018, but its job is to build genomes from scratch and do something useful with them. So we design virus genomes that hunt down cancer cells. Last year, we made 163 viruses from scratch. I love this virus too. It's called VSD, and it literally has a bullet shape. So these could very well be the magic bullets that fight. So we have just about every virus, very simple bacteria like Mycoplasma, gram-positive bacteria and gram-negative bacteria are all within reach. And now we're starting to move into the eukaryotes. And by the time we get up to 10 to the 9, which could be within 2 more decades of synthesis, genome size synthesis, genome scale synthesis, we're moving into some really interesting organisms, including us. Cost is still a factor. We've known this since we were reading genomes. It was $100 million in 2001, 20 years ago, to sequence the second genome. Today, it's about $1,000. But if you were to write this genome, write a human genome today, it would be about $100 million minimum to do that synthesis and assembly, probably more. So it's about 10 to the 6th more expensive right now to write than to read. But it will equalize, and that is a key point that you need to always hold in your head. The cost of reading and writing DNA, the cost of reading and writing a genome is going to equalize. Biofoundries are going to be a giant part of how synthetic biology moves forward in Africa, because it is very expensive to outfit and run and operate a state-of-the-art synthetic biology facility. Thankfully, you don't have to, because these foundries are coming online, these cloud labs that are going to really allow you to get up to speed much more quickly. They're standardized, highly robotic, and accessible with basically a credit card and a laptop. This will allow really you to focus on what's super important on the ground in Africa and not recreate the wheel every time. I believe that the applications that flow from synthetic biology, including energy, including medicines, etc., will have the potential to expand, much like the apps on the App Store, because we already have the countless cells ready to receive the programs that we're able to write. And AI and machine learning are going to accelerate the writing of programs. Robotics and testing and analysis is going to accelerate the testing of anything we create. So design is going to be accelerated, testing is going to be accelerated, and the computers are everywhere. So the build is everywhere. Focusing just a little bit on Africa to close. When it comes to energy, Africa is blessed. I don't recommend synthetic biology be used to make fuels, for example. You can make a new type of solar cell, that would be best. But Africa is blessed with an incredible amount of the raw materials needed for synthetic biology, for all life, which is basically sunlight and water. And the better we can utilize that energy that's clean and essentially endless and endlessly recyclable in some ways, the better we will be. So Africa has abundance. The very first efforts are already starting in Africa with companies like 54Gene to sequence, to just start sequencing the incredible diversity of life in Africa, both in the animal kingdom, the plant kingdom, the aquatic species, and of course, the citizens. This is an incredible natural resource. That data needs to be protected. And of course, any samples that are collected need to be banked. And it is just one of the richest genome banks in the world. But the absolute most important resource I've learned with synthetic biology is simply people. This is a young field, a vibrant field and dynamic, and it's never too early to start educating. I've learned this by doing it. I have a 6-year-old daughter. Her name is Ro, named after Rosalind Franklin. And she is learning not only some of the classical ways of doing biology with microscopy and puzzles, but also she's been doing state-of-the-art synthetic biology with lab robots and making art. This is a growth plate that was put on a robot. Each dot that you see there was applied by another robot. It was each color of the dots that you see here is a bacterium expressing a different designer protein that gives it a different. Bringing future, the coming future of synthetic biology and its applications. Thank you so much. I really appreciate your attention.